Hey fellow lab rats, this is Rebecca from the Lab Rat YouTube channel. In this video we're going to be discussing the staining of carbohydrates in the histopathology laboratory. Alright, let's get started. First we're going to discuss what carbohydrates are and their general functions. So carbohydrates are a very common organic compound that are composed of carbon, hydrogen, and oxygen atoms. Carbohydrates can be sugar, starches, or cellulose. Carbohydrates serve as the body's major source of energy via glucose and also comprise the structure of cell membranes in the body. They are classified by the number of carbon atoms present within the chain, the length of the chain of the carbons, and of course the location of the carbonyl group. The carbonyl group is where the carbon atom is double bonded to the oxygen. So monosaccharides are simple building block sugars. Disaccharides are composed of two monosaccharides bound together. Oligosaccharides are when there are 3 to 10 of these simple sugars linked together. And polysaccharides are when there are 10 or more of these. So glucose is a monosaccharide whose primary role is to act as a source of energy for the body. It is soluble in aqueous solutions, meaning that it is dissolvable in water. And because of this solubility, this means it cannot be seen in tissue sections. Glycogen is the storage form of glucose in the body, most specifically in the liver and also in the skeletal muscle. So what this means is if somebody consumes too much glucose for what the body needs, it gets converted into glycogen for storage. And if the body does not consume enough glucose for it to sustain itself, this glycogen that is stored in the liver and the skeletal muscle, it gets broken down into usable glucose for the body to use as an energy source. So unlike glucose, glycogen is insoluble in aqueous solution, so it does not dissolve in water. And because of this characteristic, glycogen is visible within tissue sections. The other carbohydrates that we're concerned about in the topic of histopathology are bound to either a protein or a lipid. These polysaccharides are divided into 4 groups based on their histochemical differences, and let's talk about those on the next couple slides. The first group of carbohydrates are the neutral polysaccharides. These include those polysaccharides that are glucose containing, so these are glycogen, starch, and cellulose. We've already discussed glycogen, but as a repeat, this is the storage form of glucose in the liver and skeletal muscles within animals. Starch is a primary carbohydrate in diets. Plants store their glucose in the form of starch. So glycogen is the storage form of glucose in animals, and starch is the storage form of glucose in plants. Cellulose comprises the cell wall of plants. Group 1 also contains those polysaccharides that contain N-acetylglucosamine. An example of this is chitin, and chitin is used to create a shape or a structure of organisms. So it's present in the cell walls of fungi and also present in arthropod exoskeletons. So arthropods are those little creatures like crabs, lobsters. Another example would be insects. So they have this jointed skeleton that covers their body. So this carbohydrate group gives a positive result with the PAS reaction, which stands for the periodic acid shift reaction. Now you might be saying, PAS reaction, Rebecca, I don't know what this is. We haven't talked about this before. So don't worry, it's a type of stain, and we'll get there later on in this lecture. So this group of carbohydrates gives a negative reaction with alcyon blue, colloidal iron, and musacaramine. Which again are also stains. So we'll talk about these later on in this lecture. The second group of carbohydrates are the acid mucopolysaccharides. So these are, there are 3 types or 3 categories within this group. So those that are carboxylated, those that are sulfated and carboxylated, and those that are just sulfated only. Hyaluronic acid is in the carboxylated category. This is found both in the umbilical cord tissue and also in connective tissues. In the category of carboxylated and sulfated, this contains chondroitin sulfate A, B, and C, and also heparin. So chondroitin sulfate B is present in skin primarily. It can also be in connective tissues, the aorta, and in the lung. Chondroitin sulfate C is found in cartilage, in the cornea of the eye, and also is present in the blood vessels. So heparin is found in both mast cells and in the intima of the arteries. Now, the sulfated-only category of acid mucopolysaccharides are the aorta of the human and in the cornea of cows. So all of these in this group are acidic and are PAS negative. The third group of carbohydrates are glycoproteins, which contain mucins, mucoid, mucoproteins, and mucosubstances. Just like with all these other groups, there are subcategories. So for group 3, there are neutral, carboxylated, and sulfated and carboxylated. So the neutral category contains egg whites, mucin in the stomach, and granules of the Paneth cells. And Paneth cells are specialized secretory epithelial cells located in the intestinal crypts of Leberkuhn. The carboxylated category, meaning those that have a carboxyl group, include sialoglycoproteins that contain sialic acid but no sulfate. So these sialoglycoproteins are a combination of sialic acid and glycoprotein. So these are mucins in the submaxillary gland, the small intestine, upper part of the colonic crypts, human sublingual gland, and fetal mucins. Now, the sulfated and carboxylated category are the sialoglycoproteins and colonic mucins in both humans and in sheep. Most of these are considered epithelial mucins, although some are in connective tissue. They can be PAS positive, but not always. The last group of carbohydrates, so group 4, are the glycolipids. So glycolipids are lipids that have a carbohydrate attached to them with a glycosidic bond. So these are cerebrosides and phosphatides. So cerebrosides are fatty residues that are bound to a carbohydrate. Phosphatides are non-carbohydrate containing lipids, including lecithin, cephalin, and sphingomyelin. The phosphatides are PAS positive. So what are the special staining techniques we use for carbohydrates? I've listed them here. So these are PAS reaction with diastase digestion, Mayer musicarmine, Alcian blue pH 2.5 and pH 1.0, Alcian blue with hyaluronidase, Alcian blue PAS hematoxylin, and molar Maury colloidal iron. So when we are talking about each of these special staining techniques, it's important to know the purpose of the stain. So what are we using the stain for? Its principle, so what reaction is occurring to cause it to stain? What type of tissue fixatives are required for the special staining technique, as well as what section thickness the tissue needs to be for this particular stain. When using these stains, we need to ensure that we have a quality control sample alongside to ensure that the stain is working properly. So each stain is going to have a required quality control tissue type. We'll also be discussing the reagents required for each stain, as well as the procedure. So some of these stains also have notes about them as well. So all of these things we'll be discussing for each stain type, and of course you will need to know all of this information with each of these stain types. The first of these is the periodic acid Schiff or PAS reaction. So what is its purpose? So this stain is used to see polysaccharides, neutral mucosubstances, and basement membranes. The reaction of this is based on the oxidation of tissue elements to aldehydes by periodic acid. Schiff reagent is created by treating basic fuchsin with sulfuric acid. Leucofuchsin is formed, which is a colorless compound. And following the Schiff reaction, running water washes cause the visualization of the typical Schiff color. Metabisulfate rinses are then used to remove any excess Schiff reagent, and this helps to prevent false colorization of the tissue due to oxidation of any reagent that is absorbed. In order for a tissue to be stained with a PAS reaction, it must be fixed in 10% neutral buffered formalin or Buin solution. If staining blood smears, these need to be fixed in methyl alcohol for a 10 to 15 minute period before the stain. For the PAS reaction, paraffin sections need to be cut from 4 to 5 microns in thickness. If using kidney sections, these need to be cut from 1 to 2 microns in thickness. Kidney sections are the best type of tissue for quality control for the PAS reaction. The reagents needed are 0.5% periodic acid, 1 normal hydrochloric acid, Schiff reagent, and 0.55% potassium metabisulfite. The 0.5% periodic acid is created by combining 2.5 grams periodic acid with 500 milliliters of distilled water. The 1 normal hydrochloric acid is created by combining 83.5 milliliters of concentrated hydrochloric acid with 916.5 milliliters of distilled water. The Schiff reagent is created by combining 800 milliliters of distilled water, 5 grams basic fuchsin, 4 grams sodium metabisulfite, and 80 milliliters of 1 normal hydrochloric acid. The 0.55% potassium metabisulfite is prepared by combining 2.75 grams potassium metabisulfite with 500 milliliters of distilled water. So there are two different PAS reaction procedures, the conventional procedure and the microwave procedure. For the conventional procedure, the slides must be deparaffinized and hydrated to distilled water. The sections must be placed in the 0.5% periodic acid solution for 5 minutes. And after that 5-minute period, it must be washed in distilled water using 3 total changes of that distilled water. Then it needs to be placed in room temperature Schiff reagent for 15 minutes. After this, it is optional to wash for 1 minute each in 2 jars of 0.55% potassium metabisulfite to help to remove excess stain. Then the sections should be washed in running tap water for 10 minutes. This allows it to develop its full color. The sections should then be counterstained using Harris hematoxylin with acetic acid for 30 seconds. Sections then need to be washed well to blue the hematoxylin. Dehydrate with 95% absolute alcohol, clear with xylene, and then mount it with synthetic resin. So that was the PAS conventional procedure. There is also a microwave procedure for the PAS reaction. For this procedure, the slides must be deparaffinized and hydrated to distilled water. Sections must be placed in a loosely capped Copeland jar containing 0.5% periodic acid solution. The Copeland jar is then put in a microwave oven and heated on high for 15 seconds. After this heating step, it must be washed in running water. The slides must then be placed in a Copeland jar containing room temperature Schiff reagent. The Copeland jar must be heated in the microwave oven on high for 10 seconds. And after that second heating step, it must be stirred and let sit for 30 seconds. After that 30-second period, it must be washed for 5 to 10 minutes in running tap water. After this wash, it must be counterstained for 30 seconds in Harris hematoxylin with acetic acid, then rinsed well with water. Blue nuclei in 0.75% ammonia water. You can also use other bluing reagents if needed. Then dehydrate with 95% alcohol, clear with xylene, and then mount with synthetic resin. A PAS positive result will show a bright rose color, as you can see in this photo on this slide. So this is a colon section with a small polyp that is stained with the PAS reaction and then counterstained with hematoxylin. That bright rose color, honestly kind of like the color of these slides themselves, show a positive PAS reaction. Glycogen, neutral mucose substances, certain epithelial sulfomucins and sialomucins, colloid material of the thyroid, the pars intermedia of the pituitary gland, basement membranes, fungal walls, all of these are considered PAS reaction positive. How intense a tissue stains with the PAS reaction is due to 4 different factors: the number of the available 1,2 glycol groups, the reactivity of the Schiff reagent, the structure of the polymer that is being oxidized, and the conditions in which the reaction occurs. If staining fungi, a fast green counterstain can be used instead of the hematoxylin step. If desired, the section is counterstained for 1 minute and 0.02% fast green with 1 drop of acetic acid for every 60 milliliters of staining solution. Using tap water is very important for the rinses for this procedure, as this helps for the development of the color of the stain. And again, 10% neutral buffered formalin or Buin solution are the appropriate fixatives for tissues going to be stained using the PAS reaction. Tissues cannot be fixed with glutaraldehyde because it reacts with the Schiff reaction, so just that 10% buffered formalin or Boone solution. A quality control slide must be taken through all of the steps minus the periodate oxidation step, and again, kidney tissue needs to be used for this QC material. The PAS reaction with diastase digestion is used to see glycogen present in tissue sections. This reaction is very sensitive for glycogen. Diastase and alpha amylase enzymes depolymerize glycogen into glucose and maltose molecules. Tissues that are going to be stained using this reaction need to be fixed in 10% neutral buffered formalin, absolute alcohol, or formalin alcohol. For this procedure, 2 paraffin sections need to be cut to 4 to 5 microns in thickness. One section needs to be labeled with, and the other section needs to be labeled without. The with sections are for those that will be taken through that diastase reagent. The without sections are for those that are not taken through the diastase reagent. For quality control, 2 liver sections or 2 cervix sections can be used. The reagents for the PAS reaction with diastase digestion are the 0.5% periodic acid, 1 normal hydrochloric acid, Schiff reagent, and the 0.55% potassium metabisulfite, just like the regular PAS reaction. The only difference is the addition of the malt diastase solution and a phosphate buffer with a pH of 6. The malt diastase solution is created by combining 0.1 grams diastase of malt and 100 milliliters of phosphate buffer with a pH of 6. The phosphate buffer with a pH of 6 is created by combining 8 grams sodium chloride, 1.97 grams sodium phosphate, and 100 milliliters of distilled water. The procedure for the PAS reaction with diastase digestion is as follows. So the slides must be deparaffinized and hydrated to distilled water. Now remember, we labeled one as with and one as without. So the with section is placed in preheated diastase solution at 37 degrees Celsius for a 1-hour period. The without section is placed in distilled water for this time period. Then both sections need to be then washed in running water for 5 minutes, then placed in 0.5% periodic acid solution for a 5-minute period. After that 5 minutes are up, the sections need to be washed in 3 changes of distilled water. Then the sections are placed in Schiff reagent for 15 minutes, then 1 minute each of 2 jars of 0.55% potassium metabisulfite. This step removes any excess stain from the sections. Once the excess stain is removed, the sections need to be put under running tap water for 10 minutes. Then counterstained with Harris hematoxylin with acetic acid for 30 seconds. Then the sections are placed under running water to blue the hematoxylin. The sections are then dehydrated with 2 changes of 95% and absolute alcohol, cleared with xylene, and then mounted with synthetic resin. Malt diastase contains both alpha and beta amylase, is frequently used for the digestion step of this reaction. This tends to loosen the tissue sections and doesn't always completely digest the glycogen present in the tissue. Human saliva only contains the alpha amylase enzyme, so this can be used to digest the glycogen when put on tissue for 20 minutes at room temperature. Now the photo on the left-hand side of the slide here shows a fatty liver section that is stained using the PAS reaction with hematoxylin or PASH stain. This is not digested. The photo on the right-hand side is the same tissue that is stained with the PAS reaction with hematoxylin and then has been digested with the diastase. The next thing we're going to be discussing is Mayer mucicarmine. So this is used for the staining of epithelial mucin within the tissue. For this stain, aluminum forms a complex with carmine, and this complex then binds to the acid groups of mucin. Tissues need to be fixed in 10% neutral buffered formalin, and the sections need to be cut to 4 to 5 microns in thickness. Quality control tissue for Mayer mucicarmine can be colon tissue, tissue from the small intestine, or appendix tissue. The reagents needed for the Mayer mucicarmine stain are mucicarmine working solution. So this is created by combining 10 milliliters stock solution with 40 milliliters of distilled water. Weigert iron hematoxylin. And so to prepare this, there are solution A and solution B. And solution A is created by combining 10 grams of hematoxylin and 1,000 milliliters of 95% alcohol. Solution B is created by combining 475 milliliters of distilled water, 5 milliliters of hydrochloric acid, and 20 milliliters of 29% ferric chloride. Now, equal parts of solution A and solution B need to be mixed to create the Weigert iron hematoxylin. Then there is a 0.25% metanyl yellow solution. So this is prepared by combining 0.25 grams of metanyl yellow, 100 milliliters of distilled water, and 0.25 milliliters of glacial acetic acid. So the procedure for the Mayer mucicarmine stain is as follows. So deparaffinized tissue sections that are hydrated to distilled water. The tissues need to be stained in working Weigert solution or working Weigert hematoxylin for 7 minutes, then wash in running water for 10 minutes. After this 10-minute period, the tissue sections need to be stained with mucicarmine for 1 hour, afterwards rinsing and then removing the excess water. Then the tissue is stained in metanyl yellow for 30 seconds to 1 minute in length. The tissue then needs to be dehydrated with 3 changes of 95% alcohol and 3 changes of absolute alcohol. Then cleared with xylene and mounted with synthetic resin. With the Mayer mucicarmine stain, mucin will be a deep rose to red in color. Cryptococcus neoformans will also be a deep rose to red in color with this stain. So Cryptococcus neoformans is a fungus that lives in the environment. So people, usually those who are immunocompromised, can become infected with it, often in the lungs, and it can occur in the brain as well. Nuclei will be black with this stain, and other tissue elements are blue or yellow. So let's look at some examples of what this stain looks like here on the next 2 slides. So this photo on the left-hand side of this slide shows a small intestine section that is stained with Mayer mucicarmine. So these red goblet cells here, I'll go ahead and circle them here a little bit on this slide. All right, so these are easily identifiable because they are mucin. So nuclei are also present here in this black color. So here's all the nuclei. Okay, those are nuclei in that black color. Now the photo on the right-hand side of the slide shows a small intestine section that has been overstained in the metanyl yellow staining step. It masks the mucin, so you can't really see it here. You can see the difference between the two. The photo on the left-hand side of this slide shows an autolyzed small intestine section. So as you can see, there are no deep red rose to red color on this slide because no goblet cells are present. Autolyzed tissue is not acceptable for QC for the Mayer mucicarmine stain. Now the photo on the right-hand side of the slide shows a section stained with Mayer mucicarmine that shows Cryptococcus neoformans. So these deep red, a deep rose color, or red color are the Cryptococcus. Alcine blue with a pH of 2.5 is used to stain acid mucopolysaccharides. Alcine blue is a dye that is soluble in water and blue in color. When used in a 3% acidic acid solution, which gives it a pH of 2.5, alcine blue stains both sulfated and carboxylated acid mucopolysaccharides and sialomucins. Tissues need to be cut 4 to 5 microns in thickness and be fixed in either 10% neutral buffered formalin or Buin solution. Quality controlled tissue for alcine blue with a pH of 2.5 can be tissue from the small intestine, colon, or the appendix. The reagents needed for the alcine blue pH of 2.5 stain are 3% acetic acid solution. This can be prepared by mixing 15 milliliters of glacial acetic acid and 485 milliliters of distilled water. 1% alcine blue solution, which is created with a combination of 5 grams of alcine blue 8GX and 500 milliliters of 3% acetic acid. Adjust that pH to 2.5, filter, and then add a few thymol crystals. And then the other reagent is nuclear fast red solution, which is created from the combination of 0.5 grams of nuclear fast red, 25 grams of aluminum sulfate, and 500 milliliters of distilled water. The procedure for this stain is as follows. Deparaffinize in hydrated distilled water. Slides are put into 3% acetic acid solution for 3 minutes, then put in Alcyon blue solution for 30 minutes at room temperature, or 15 minutes in a 37°C water bath. After that step, they are rinsed in 3% acetic acid, then washed in running tap water for 10 minutes. Then they are rinsed in distilled water. The sections are then counterstained with nuclear fast red for 5 minutes and then washed for 1 minute in running tap water. Then the tissue is dehydrated with 2 changes of 95% alcohol and absolute alcohol, cleared in xylene, and then mounted in synthetic resin. With the Alcyon blue pH 2.5, weakly acidic sulfated mucose substances, hyaluronic acid, and sialomucins will be dark blue in color. The background will be pink to red. We'll look at a picture examples of this in just a couple minutes here. Let's talk about the Alcyon blue pH of 1.0 first. Alcyon blue at a pH of 1.0 is used to stain sulfated mucose substances. When combined with the 0.1 normal hydrochloric acid with a pH of 1.0, Alcyon blue stains only sulfated acid mucopolysaccharides and sialomucins. The carboxylated versions are not stained with the Alcyon blue with a pH of 1.0. Tissue needs to be cut from 4 to 5 microns in thickness and fixed in 10% neutral buffered formalin and Buin solution. I honestly really should have said or Buin solution. So the tissues need to be fixed in either 10% neutral buffered formalin or Buin solution, not both. Quality control for this is the same as the Alcyon blue pH of 2.5, which is small intestine tissue, appendix, or colon tissue. The procedure for the Alcyon blue at a pH of 1.0 stain is as follows. Deparaffinize, hydrate to distilled water, then rinse in 0.1 normal hydrochloric acid and stain in 1% Alcyon blue and 0.1 normal hydrochloric acid for 30 minutes at room temp, or 15 minutes in a 37-degree water bath. After this step, the slides are rinsed with 0.1 normal hydrochloric acid. The sections are blotted with filter paper. It's important that it is not washed with water, as it can cause a pH change, which leads to the stain not working properly. So after this blotting with the filter paper, the section is counterstained with nuclear fast red solution for 5 minutes and then washed in distilled water. Then dehydrate with 2 changes of 95% and absolute alcohol, clear it with xylene, and then mount with synthetic resin. Sulfated mucose substances stained with Alcyon blue with a pH of 1.0 appear as a pale blue color. The background is pink to red in color. We'll see that on the next slide. Alright, so the photo on the left-hand side of the slide is a section of colon with a small polyp that is stained with the Alcyon blue at a pH of 2.5. Carboxylated and sulfated acid mucopolysaccharides and sialomucins, also called glycoproteins, stain at this pH. Now the photo on the right-hand side of the slide is the same tissue section that is stained with Alcyon blue at a pH of 1. Only sulfated acid mucopolysaccharides and sialomucin stain at this pH. Alcyon blue with hyaluronidase is used to differentiate between epithelial and connective tissue mucins. We've already discussed the principle and methods of the Alcyon blue stain, so I'm not going to repeat them here. So now with the Alcyon blue with the hyaluronidase, this uses this enzyme to digest connective tissue mucin and leave the epithelial mucins untouched. Tissues should be fixed in 10% neutral buffered formalin for this stain. For this stain, 2 paraffin sections cut at 4 to 5 microns in thickness should be used. One should be labeled in quotes with digestion, and the other should be labeled in quotes without digestion. For quality control for this stain, 2 slides with umbilical cord tissue should be used, and of course one that is digested and one without digestion. Small bowel tissue tissue, colon, or appendix can be used as a second control if indicated to demonstrate epithelial mucins. The reagents for the Alcyon blue with hyaluronidase stain are 0.1 molar potassium phosphate. This is prepared by mixing 13.61 grams of potassium phosphate with 1,000 milliliters of distilled water. 0.1 molar sodium phosphate, which is prepared by combining 14.2 grams of sodium phosphate with 1,000 milliliters of distilled water. Buffer solution with a pH of 6, so this is prepared by mixing 94 milliliters of potassium phosphate with 6 milliliters of sodium phosphate. The hyaluronidase digest solution, so this is prepared by combining 50 milliliters of a buffer solution to 25 milligrams of testicular hyaluronidase. Alcyon blue staining solution, which is prepared by combining 1 gram of the Alcyon blue 8GX with 100 milliliters of 3% acetic acid, and it needs to be adjusted to a pH of 2.5, and thymol crystals needed to be added. And then also a nuclear fast red solution is needed for this as a reagent for this stain. The procedure for performing the Alcyon blue with hyaluronidase stain is as follows. Preheat a Copeland jar of buffer and digestion solutions at 37 degrees Celsius for 1 hour. So you want 2 here, so 1 of just the buffer solution and 1 of the digestion solution. You want to deparaffinize the tissue and hydrate it to distilled water. So sections that are labeled with digestion need to be in the preheated digestion solution in the Copeland jar, and then the sections that are labeled without digestion need to be put in the preheated buffer. So again, you want to have these in the with digestion in the digestion solution, the without digestion in the buffer, and you want to have these sit for 2 hours in those Copeland jars. After that 2-hour period, you want to wash with running water for 5 minutes, and then place them in 3% acetic acid solution for 3 minutes. Then it needs to be stained in the Alcyon blue stain for 30 minutes. After the Alcyon blue step, you want to have it a 10-minute running tap water rinse, and then a distilled water rinse after that. And then you want to counterstain with a nuclear fast red for 5 minutes, and then put the slides under running tap water for 1 minute. After that running tap water step, you want to dehydrate with 2 changes of 95% alcohol and absolute alcohol, and then clear it with xylene. The photo on the left-hand side of the slide here shows an umbilical cord tissue that is stained with Alcyon blue with a pH of 2.5, and this one on the left has not been digested. The photo on the right-hand side of the screen shows the exact same tissue that is stained with the Alcyon blue with a pH of 2.5 that has been digested with testicular hyaluronidase. So note the differences here. One's digested and one is not. Alright, so there's also an Alcyon blue PAS hematoxylin stain, and this is used to differentiate between neutral and acidic mucosubstances. Acidic mucosubstances are stained with the Alcyon blue, and the neutral mucosubstances stained with the PAS reaction. Tissues must be fixed in either 10% neutral buffered formalin or Zanker solution for this staining reaction. The sections must be cut from 4 to 5 microns in thickness unless you're using tissue from the kidney. Kidney sections should be cut at 2 to 3 microns in thickness. Quality control for this stain can either be a kidney or mucin control. The reagents needed for the Alcyon blue PAS hematoxylin stain are 3% acetic acid solution, and this is prepared by combining 3 milliliters of glacial acetic acid with 100 milliliters of distilled water. Alcyon blue at 2.5, which we've already discussed how to prepare. 0.5% periodic acid, which is prepared by combining 2.5 grams of periodic acid and 500 milliliters of distilled water. A stock reducing rinse, which is prepared by combining 10 grams of sodium metabisulfite with 100 milliliters of distilled water. A working reducing rinse, which is prepared by combining 2.5 milliliters of stock reducing rinse with 50 mils of distilled water. And then lastly, Schiff reagent is required for this stain. The procedure for the Alcyon blue PAS hematoxylin stain is as follows. So the tissue is deparaffinized and brought to water. It is put in 3% acetic acid for 1 minute and then stained with the Alcyon blue stain for 30 minutes. After this Alcyon blue step, it will need to be washed in running tap water and then rinsed with distilled water. Then it needs to be put in 0.5% periodic acid for a 10-minute period and then placed under running tap water for 5 minutes. After the running tap water, it then needs to be rinsed with distilled water. Then the sections will need to be in shift reagent for 10 minutes and then another 10-minute period in a reducing rinse. Harris hematoxylin with acetic acid is an optional step, but if needed, this should be in there for about 1 minute. The section should be in there for 1 minute. After that, you want to place the sections in running water for 10 minutes, and after that 10-minute period, the tissue needs to be dehydrated with 2 changes of 95% in absolute alcohol, cleared with xylene, and then mounted with synthetic resin. This photo shows a cervix tissue section that is stained using the Alcyon blue PAS hematoxylin technique. Glycogen is present here with the stratified squamous epithelium, which you can see here. All right. It's stained a rose color by the Schiff reagent. The mucin secreting gland is a dark blue to purple color here. All right. This indicates there is a mixture of both neutral and acidic mucins. The Muller-Maury colloidal iron stain is used to show carboxylated and sulfated mucopolysaccharides and glycoproteins. And now carboxylated and sulfated mucosubstances absorb colloidal ferric ions at a low pH. Excess reagent is washed out and Prussian blue is used to show any iron that is bound to this tissue sample. So tissues need to be fixed in 10% neutral buffered formalin, an alcoholic formalin, or Carnoy solution. Chromate fixatives should not be used for this stain. For this stain, tissues need to be cut at 4 to 5 microns in thickness. QC or quality control can be small bowel tissue, appendix tissue, or colon tissue. Reagents for the Muller-Maury colloidal iron stain are 29% ferric chloride, so this is created by mixing 29 grams of ferric chloride with 100 milliliters of distilled water, a molar colloidal iron stock solution, a working colloidal iron solution, which is prepared by combining 10 milliliters of the stock colloidal iron solution with 18 milliliters of distilled water and 12 milliliters of glacial acetic acid. 2% potassium ferrocyanide solution, which is created by combining 2 grams of potassium ferrocyanide and 100 milliliters of distilled water. 2% hydrochloric acid, which is prepared by combining 2 milliliters of hydrochloric acid with 100 milliliters of distilled water. Ferrocyanide hydrochloric acid, which is prepared by combining 25 milliliters of 2% potassium ferrocyanide with 25 milliliters of 2% hydrochloric acid. A 12% acetic acid solution, which is prepared by combining 24 milliliters of glacial acetic acid with 176 milliliters of distilled water. And lastly, a nuclear fast red solution. For the Muller-Maury colloidal iron stain, there is a conventional procedure and a microwave procedure. The conventional procedure is as follows here. So you want to deparaffinize the tissue and hydrate to distilled water. It needs a brief 12% acetic acid solution rinse and then placed in a colloidal iron solution for 1 hour. After that 1 hour of colloidal iron solution, it needs to be rinsed in 3 changes of 12% acetic acid solution at 3 minutes each. So 3 changes at 3 minutes each. Then it needs to be immersed in ferrocyanide hydrochloric acid solution at room temperature for 20 minutes. And after that 20-minute period, it needs to be washed in running tap water for 5 minutes. Then counterstain in the nuclear fast red stain for 5 minutes. And after that 5-minute period, it needs to be rinsed with running water for 1 minute, and then dehydrated, cleared in xylene, and then melted with synthetic resin. Now, the microwave procedure, so the previous one was the conventional procedure, this is the microwave procedure for the Muller-Maury stain, is as follows. So again, you want to deparaffinize the tissue and hydrate to distilled water. It needs a 1-minute rinse in 12% acetic acid solution. And then after that period, there will be a colloidal iron solution in a glass Copeland jar. You want to put the sections in there. Microwave at 60 watts for 3 minutes, and you want to dip up and down those slides in the warm solution for 10 minutes. Then rinse in 4 changes of 12% acetic acid solution, each of those at 1 minute. So 1 minute rinses in 4 different changes of that 12% acetic acid solution. After that, you want to immerse in ferrocyanide hydrochloric acid solution, microwave again at 60 watts for 2 minutes. And again, you want to dip up and down after this period for 2 minutes with the slide still in that warm solution. Then run it in tap water for 1 minute, and then rinse it in 2 changes of distilled water. After that distilled water step, it needs to be counterstained in nuclear fast red for a period of 3 minutes, and then again rinsed in running water for 1 minute, and then dehydrated, cleared with xylene, and then melted with synthetic resin. Acid mucopolysaccharides and sialomucins will stain a deep blue with a Muller-Maury stain. Nuclei will be pink-red in color, and the cytoplasm will be pink. This stain does give a deeper, more intense stain than the Alcian blue does, although it is not considered specific for acid mucopolysaccharides. Muller-Maury stain is a great stain for Cryptococcus neoformans, which we have already discussed within this lecture. The photo on the left-hand side of the slide is a lung tissue section that is stained with the Muller-Maury colloidal iron stain. The cartilage, goblet cells, and mucin-secreting gland are strongly stained here. Now the photo on the right-hand side is the same colon tissue with small polyp that we've seen throughout this lecture, this time stained with the Muller-Maury colloidal iron stain. The photo on the left-hand side of the screen shows a microwave colloidal iron stain showing Cryptococcus neoformans. And again, we've discussed this fungus previously within this lecture, so I'm not going to talk about it again. The right-hand side photograph shows a small intestine tissue that is stained with the Muller-Maury colloidal iron stain and counterstained with hematoxylin. So this is an example of when fresh stain is not used for this procedure, noted by the marked non-specific background staining. So this stain needs to be repeated. All right, so this wraps up our lecture on carbohydrate staining within the histopathology laboratory. If this video helped you, please make sure to like it and subscribe to my channel for more educational laboratory content. And as always, please leave your questions in the comments section. I'll be happy to answer any questions about this lecture or, of course, any other laboratory material. Until next time.